Hello and welcome to the Here Exposomics webinar series. Today's webinar is on racial disparities in chemical exposures. Thank you all for joining us. This is a very exciting topic. Today we are talking about how and why chemical exposures may differ among racial groups and how that may impact human health. This is a very important question for environmental justice communities that are experiencing differential exposures. I am Dr. Susan Lessig. I'm a program officer with the NIH ECHO program, Environmental Influences on Child Health Outcomes, whose goal is to enhance the health of children for generations to come. This webinar is part of the Here Exposomics webinar series that began over 2 years ago to inform the wider research community about the latest analytical approaches for characterizing the exposome, or the totality of exposures throughout the lifetime. The webinars showcase how new methods can be incorporated into a variety of types of human health studies or environmental health studies. The ECHO program has partnered with HERE for the last 8 years to perform a variety of targeted and untargeted assays using the existing ECHO cohort biospecimens for the program. Today we have two incredible speakers to share with us their work on perfluorinated chemicals and human health. First, we have Dr. Shelley Liu, who's an associate professor in the Department of Population Health Science and Policy at the Icahn School of Medicine at Mount Sinai. She's a data scientist and biostatistician working at the intersection of precision, environmental health, metabolic and cognitive health, with an emphasis on health equity and policy. Please join me in welcoming Dr. Liu.
Hi, everyone. Thank you so much for attending this talk today and taking time out of your busy schedule to be here. So today I'll be talking about our work developing the US PFAS exposure burden calculator for cross-study harmonization and how we can use that to do things like uncover racial ethnic exposure disparities, identify vulnerable populations for interventions, and in particular, how this calculator helps us develop a fair and standardized and nationally representative scale to quantify kind of the totality of exposure burden to PFAS. And so the big premise of my talk today is, rather than focusing on the individual PFAS and what might be the drivers of health outcomes, to think about PFAS as a chemical class and think about the totality of exposure to this chemical class and how do we best measure that? Because we know that PFAS are encompassing thousands of chemicals, depending on what definition you're using. And we do not have analytical chemistry tools to be able to measure concentrations of all those PFAS. So we're conceptualizing your PFAS exposure burden as sort of this latent variable. And then we're using methods from psychometrics and the development scales to be able to then estimate using the PFAS analytes that we do measure in different biospecimens to be able to estimate your underlying exposure burden to PFAS as a chemical class. And so having that estimate of total PFAS burden, we can use it for different things, including for biomonitoring, environmental justice, identifying disparities, and also if you're interested in creating disease risk calculators to have a simple summary metric to be able to use there. And then I'll talk about how we're refining this calculator to also account for exposure source heterogeneity. So, we know that we have a very diverse population in the US. Different people have different diets and behaviors that expose them to different sets of PFAS. And so we want to make sure that our scale is taking all that into account as well. And so that's kind of the big picture of what I'll be talking about today. And we'll show how we used our calculator to then identify racial ethnic disparities, particularly finding that Asians have higher PFAS exposure burden than whites and other racial ethnic groups. And in particular, low-income Asian Americans have really much higher PFAS burden compared to low-income groups of other race ethnicities. So that's kind of the big picture summary of what I'll be talking about. Okay, so to begin with, I think this is a graphic from Diabetes Care Journal, talking about the paradox of progress, how we can see that over the last few decades, there's been a significant increase in synthetic organic chemical production. And that really tracks with like an increase in other chronic diseases like diabetes. And so today, this is going to be about PFAS, a specific class of synthetic organic chemicals where most everyone unfortunately has detectable levels of PFAS chemicals in their blood because they're so pervasively used and as oil, water, repellents, and industrial and consumer products. And so, you don't have to work in an occupation to be exposed to these chemicals. We're just exposed to many of them in daily life. So they're linked to a variety of health outcomes like cancers, elevated cholesterols, impaired immune system functioning, and others. And so, when we think about modeling the PFAS exposome, we might be interested in different scales of resolution of looking at PFAS. So on one hand, that there's potentially thousands of chemicals. Perhaps we can't identify thousands in our own blood, but we can identify maybe at least a few dozen. And so we might be, for example, interested in the individual ones, but there might be other cases where we're really interested in getting a good measure of the totality of your PFAS exposure burden as a good summary metric to be used in other downstream analysis. And so the challenge, though, is that, although there has been an exponential increase in the number of EPI studies on PFAS, so we see from, early 2000s, there were basically no studies, almost 0 or 1. And then 2021 now, there was 123 PFAS EPI studies published. There's no standardized common scale of PFAS exposure burden. So some studies, they'll use the summed PFAS as a proxy of their total PFAS. And so what they do is they might just add up the concentrations of all the PFAS analytes that they have to measure. The problem there is that different studies are measuring different sets of PFAS, different labs are measuring different sets. And so there's no way to put things onto a common scale. And especially having a common scale will help with cross-study harmonization and doing pooled analysis, meta-analysis, especially for underrepresented minorities who you may need to pull together data from different studies to get a large enough sample size to study them. And so we've seen that there's definitely a need for looking at total PFAS as a chemical class. For example, the recent National Academies of Science, Engineering, and Medicine, NASEM, put a guidance on clinical biomonitoring, where they suggest using a sum of 7 PFAS analytes as that proxy of total PFAS. And so I think that's great and really shows a need for a summary metric to this class of chemicals. But at the same time, we can think about that there's thousands of potentially PFAS chemicals. And so that sum of 7 may be a really great proxy of total PFAS for, potentially a lot of your population. But there are other people who have potentially very different exposure sources to PFAS. They may not be really exposed to those 7 PFAS that you're including in the total proxy. And so they might look like they might artificially look like they have low PFAS if you're using that proxy of the 7 as your total exposure. But they actually might have pretty high levels of some other PFAS analytes that you're not measuring or you're not including in that total. So we just want to, use other data science and psychometric methods to really think about, how we're creating summary scores, making sure that we're creating fair summary scores that are informative for all people. And perhaps it's true that some of these sums can do really well, but then perhaps also there are cases where some more advanced methods could really be helpful. So thinking about how to set a standardized common exposure scale across studies, and then also account for the fact that many labs will measure different sets of PFAS analytes. And so some of them are in common and some of them are quite different between labs. And so using methods that can allow us to make full use of all of the measured data, not just the ones that are in common across studies. And then also thinking about how different subpopulations could have different diets and behaviors that would systematically expose them to different sets of PFAS. So accounting for the exposure source heterogeneity. And so, in the big picture, when you have these exposure scores, you can use them for developing precision interventions. For example, identifying racial ethnic disparities in exposure, and then being able to more precisely target your interventions, particularly in primary prevention of disease. So we have used item response theory as an alternative to creating summary scores for PFAS as compared with some concentrations. So these are a set of really well-developed methods, psychometric methods, used in the educational testing literature for measurement and the development of scales. So on the surface, it seems like it has nothing to do with what we're interested in. In the educational testing world, they're interested in estimating someone's latent cognitive ability. So for example, their math ability. And what they'll do, they'll give a set of, for example, like the GRE or the ACT, they'll give a set of test items to a lot of people and use their responses on those test items across a very diverse population to then calibrate a scale. And so then they'll use, how many you got right, as well as your pattern of responses to those items to then estimate your latent math ability. And in that way, we can actually bring it over to the environmental exposure space and think about the different PFAS analytes that we measure in serum and plasma as these items that inform something about your latent PFAS exposure burden. And so your PFAS exposure burden is a latent unknown variable that we're trying to estimate. And so through both the concentrations of the individual PFAS as well as your exposure patterns, we can estimate that using item response theory methods. They're kind of akin to nonlinear factor analysis. There's some mathematical overlap there. So I'm not going to get into the mathematical details, but these papers I'll be talking about are published. And so there's a lot more details there if you're interested, as well as code if you're interested in that. And so what we created was a 2017-2018 US PFAS exposure burden calculator, this R Shiny app that enables a common scale of burden scores across studies. So you can go to our website and use this. And so you basically can input, if you have PFAS analyte data measured on participants, you can input their concentrations into our calculator. And ours is calibrated on nationally representative data from NHANES. And so we'll give you a PFAS burden score now for each participant in your data. And so those scores can be interpreted relative to the national US population. So they're standard normal. So if you have a score of 0, it can be interpreted as kind of this participant as kind of the average PFAS burden of the US population. If they have a score of 1, their score is 1 standard deviation above that of the US population. And so there are these continuous scores that we're going to provide. And so it'd be really interested in trying, definitely try it out. And it can be used in conjunction with summed PFAS analytes as another way to just create a proxy. And what's also really great about these is that we've shown that after we've calibrated our calculator, it's okay if you don't measure the full set of the PFAS analytes that we have. You can still place your participants' data onto our scale, and we will still provide valid and informative PFAS burden scores for your participants. And so this is something that's really well known in the educational testing world. So when they use item response theory for educational testing, they will, for example, for the GRE, they're not going to give the same test form to different people because they want to maintain test integrity. So they've already calibrated all their items. And so they'll give different sets of tests to different people, and then they'll put everything back onto the same scale using anchor items. And so that's what we have done here too, is that, for example, we did a sensitivity analysis where we already had a calibrated calculator, and then we placed PFAS, we artificially created a scenario where people are missing PFAS biomarker data on some of them. And we put it back into our calculator to estimate the PFAS burden scores. And we see that even if you're missing some biomarker data, we can still estimate basically the same associations with health outcomes as if you had the full set measured. And so we've also done a demonstration of how to use this calculator in epi studies and comparing epi studies from 2 different studies of child health. And so then we are refining it even further by considering exposure source heterogeneity when estimating PFAS burden scores. So particularly thinking about how we know that there were different population subgroups that might be exposed to different sets of PFAS, but these are not really well known. The exposure source heterogeneity can be complex. It can be a complex combination of diet and behavior and sociodemographic factors. And so then we thought that perhaps there could be systematic differences in exposure sources. And so you might need customized scoring algorithms for PFAS burden to ensure that the quantification of burden is equitable and informative for all people. And so we thought if you just use a one size fits all approach, so for example, thinking that the sum of 7 PFAS is a good proxy for total PFAS for all people, that might be true for a large segment of your population, but it might not be true for everyone. And so you might end up underestimating PFAS burden for some people. And then you might also, as a downstream result of that, end up masking disparities in exposure burden across population subgroups. So we used another version of item response theory called mixture item response theory. And we wrote a paper talking about how this could be used to create a customized exposure burden score to PFAS mixtures to enable more equitable comparisons across population subgroups. And so the basic premise here is that rather than a one size fits all approach for estimating PFAS burden for everybody, as the IRT method that we previously discussed in our calculator does, we thought that there could be, you could do a data-driven approach where you could actually identify different latent classes that necessitate their own scoring algorithms. But at the same time, you still want all the scores to be comparable. So then we use what are called anchor items to place everything back under the same scale. And so we can think of these latent classes. We identified in our NHANES data 4 latent classes that we can think of as like different exposure source profiles that they're so different that they necessitate different scoring algorithms for quantifying exposure burden. And so now your score is a weighted combination of these scores from these different algorithms. And it's weighted based on how well you yourself fit that profile. And so essentially, we identified 4 complex exposure source profiles that need distinct scoring algorithms. So for example, one of them is characterized by people with higher SCS. Another one is characterized by people with lower SCS, lower food security. Another one characterized by males, whites who are of older age. And so we see that it's not just a single, these exposure source heterogeneities are pretty complex. And so now we can get more customized scores that can take into account exposure heterogeneity. And so we wanted to see then, essentially, if we can find out anything more in terms of racial ethnic disparities in exposure burden. So when we looked at, and our main data was NHANES data from 2013 to 2016, and our validation was a later data set from NHANES, we can find that really, if you compare our mixture IRT scores and the NASEM sum scores in terms of looking at disparities against a reference group of non-Hispanic whites, we can see very consistently that Asians have significantly higher PFAS burden than whites. And so this is kind of borderline significant when we're using the NASEM sum of just the 7 in our main data, but it's null when you use the validation data. So it just shows that the disparities are more apparent and consistent when using mixture IRT scores compared with the NASEM sum. And we can pretty consistently see that Blacks don't have significantly different exposure burden compared to whites. Hispanics and those of other race or multi-race groups have actually lower exposure burden compared to non-Hispanic whites. And I think the story gets really interesting when you look at segmenting this out by socioeconomic status, that particularly for low income Asian Americans, so those with an income family income to poverty ratio of less than 1, we actually see that Asians have significantly higher exposure burden to PFAS compared to whites who are also low income. And that disparity sort of goes away once you get into the higher incomes. And so this is pretty consistent when you're using the mixture IRT scores or the NASEM sum scores. And we're also seeing this disparity in the low income groups in other validation data as well in NHANES. So, and this in general, this disparity in terms of Asians having higher PFAS burden has been seen in other data as well. For example, in the California biomonitoring data. So it's definitely just something really important to research further and also identify what might be those exposure sources that are contributing to this disparity so we can develop some more precision interventions. And so there are a couple of takeaways. One is that if you do PFAS epidemiology or PFAS exposure science work, definitely encourage you to try out our PFAS exposure burden calculator to get PFAS exposure and burden scores for your participants, which are normed to the US nationally representative data. And then our work in refining this calculator further suggests that biomonitoring and risk assessment may want to consider using a summary PFAS exposure metric that can also account for exposure heterogeneity so that we can just make sure that when we're using a summary metric that it's fair and informative for all people. And we're also working on extending our PFAS burden calculator to include more PFAS analytes and create a harmonized calculator over decades. So trying to account for this change in time trends and regrettable substitution. So this idea that, your legacy PFAS concentrations in your serum might be going down, but then actually your serum concentrations of replacement PFAS might be going up. And so your PFAS burden actually might be remaining the same over time. And so the thing is, if you're only looking at, if your summary metric is, for example, mostly focused on legacy PFAS, you might end up over time underestimating your total PFAS burden. And so that's where we're trying to work on further. And so if you have PFAS data that you'd be willing to share to help calibrate our PFAS burden calculator, especially we're always looking to augment it to be even more racial and ethnically and geographically diverse with more PFAS analytes and with non-targeted PFAS data, please contact me. And so these are some references. The papers I talked about today, the more detailed statistical methods as well as code are in the papers. Yeah, these are my acknowledgements of my wonderful collaborators as well as funding sources. And I'm happy to take any questions. Thank you very much.
Thank you so much for that talk, Dr. Leah. That was so many interesting facets of your work. Really appreciate hearing about it. We have time for some questions now for Dr. Liu. If there's anyone who has questions, I believe you can put them in the Q&A. Not sure if it's open for raising hand or, so please. Okay, I see a question from an attendee who says, does your calculator account for where the PFAS concentration testing is coming from? For instance, blood, serum versus tissue?
Yeah, that's a great question. Right now, because of the data that we have, we're using serum. And so I'm definitely interested in developing for other tissues as well. And if you have data that you'd like to share, please contact me.
Great, thank you. It may be very of interest to ECHO investigators who have some data on PFAS as well. Another question from Alex Sitarik. Are you developing different scores for different ages, adults versus children?
Yeah, that's a great question. I think in the next iteration, I think we're going to work on this. Yeah, because it would be, I agree, it would be really interesting to create sort of norms based on different age groups, especially since we know that, older people just, because of how PFAS can accumulate in the human body, older people tend to have higher PFAS.
Great, thank you. Another question from the Q&A is, was there any further exploration of source attribution? Do you know about what is contributing to higher concentrations in some populations?
Yeah, that's a great question. We did do some exploration. Of course, we're limited, for example, to the type of data that's available in NHANES. So, for example, I think fish consumption can be a major contributor of PFAS, but the way that dietary data is collected in NHANES is not so refined in really knowing like how often you're eating fish or what types of fish. So I think the extent of having even more detailed data would be great. And, also, for example, water sources. I think there's a lot of need for additional exploration on this front.
Yes, I completely agree that source attribution of chemicals is an open area needing that much more work. There's another question here. Are the scores mainly developed in healthy individuals, or have you looked at different groups of individuals with varying health conditions, cancer survivors, or other diseases?
Yeah, that's really interesting. I would say because they're calibrated on NHANES data and nationally represented data, I think NHANES participants do have some chronic diseases, but they probably tend to be more healthy because you have to be able to go to the NHANES site and like do all those assessments. So yeah, I think it would be great to look at more diverse individuals with different disease states, etc.
Mm-hmm. I'm not sure if you have the ability to look at, to do a case cohort kind of study in NHANES, but in ECHO, there's a lot of outcome data as well as the exposure. So
okay, great.
Curious as to how you would see this fitting into an epidemiology study.
Yeah, that would be super interesting. I'll have to chat further.
Sure. I'll ask one final question. The methodology can you use your method to look at other groups of contaminants? For example, volatile organic contaminants, or have you already done that?
Yeah, I think that would definitely work for other groups of contaminants as well. We have mostly focused on PFAS, have done a bit of work for phthalates as well, but I think we were just the first to really bring item response theory from the psychometrics world to environmental epidemiology. So I think there's a lot of potential for this class of methods to advance these fields. We'd be happy to chat further if I can be of help as well.
Thank you so much for sharing that with us today. And we will, if you have any follow-up, Dr. Liu will be happy to talk to you. All right, our next speaker today is Dr. Donghai Liang. He is an assistant professor at Emory University in the Rollins School of Public Health, Gangarosa Department of Environmental Health. He's an exposure scientist and molecular epidemiologist who's interested in characterizing the human exosome and elucidating molecular mechanisms of complex human health responses to diverse environmental exposures. He has received several early career awards, including an ECHO Early Career Award, the Opportunities and Infrastructure Fund. So Dr. Liang will talk to us about metabolomics investigations on PFAS and fetal growth in African American mothers and children. Go ahead, Dr. Liang.
Thank you so much for the great introduction. I hope that everybody can see my screen. Hi, good afternoon, everybody. I'm very happy to be here. And as our chair said, I'm a assistant professor from Emory University. And today I'm very happy to talk about a recent metabolomics investigation, really to look at the impact of prenatal exposure to PFAS on fetal growth among African American. So our study was supported by the ECHO program as well as with the great support from the HERE program. So as Dr. Liu just said, many of you already know about PFAS. So this per and polyfluoroalkyl substance, also we call them forever chemicals, are really a group of widely used and long-lasting chemicals, and they break down very slowly over time. And there's growing evidence in the recent decades that exposure to PFAS can lead to numerous adverse health effects, and this include negative birth and children's health outcomes. At the same time, this public health burden is not shared equally across the population, with communities of color and those are at the lower socioeconomic status, as well as minority populations such as African American, pregnant people and children that are disproportionately exposed to higher environmental exposure and at the same time experience higher risk of reduced fetal growth and other adverse birth outcomes. And despite this epidemiological evidence, the underlying mechanisms linking PFAS exposure and adverse birth outcomes are very complex and largely unknown, which limit the development of effective interventions to protect the vulnerable population. So with the development of high throughput sequencing and analytical technology, many omics-based approaches have emerged as promising platforms that can potentially tackle these challenges by exploring the underlying mechanisms. And these omics technology are increasing being used in exposomics research. And for my talk today, I will mainly focus on metabolomics, which is the global detection and characterizations of small molecules called metabolites in the human body. And compared to other omics approach, metabolomics has a number of theoretical advantages. The metabolome is the final downstream product of the gene transcription and gene environment interaction, and therefore the changes in the human metabolome are amplified relative to changes in the genome, epigenome, transcriptome, and the proteome. And additionally, as the downstream product, the metabolome is the closest to the phenotype of the biological system. So to further address the knowledge gaps underlying the impact of PFAS exposure on maternal and child health, we designed and conducted the PFAS exposure and child health, the PEACH study, which is supported by the ECHO OIF funding as well as the NIH HERE program. So in the PEACH study, we aim to use targeted PFAS exposure assessment and high-resolution metabolomics to investigate the biological mechanism underlying the impact of exposure to PFAS on fetal growth among about 300 participants within the Atlanta African American maternal and child cohort. So thanks to the Emory Cheer Laboratory led by Drs. Dana Barr and Barry Ryan, as well as the NYU and Wadsworth Cheer and HERE Laboratory led by Dr. Cannon, the serum concentration of 12 PFAS were quantified using targeted exposure assessment in over 300 participants, where we look at their early pregnancy and late pregnancy serum sample, as well as more recently working with Dr. Cannon's team, we were able to assess dry blood spots concentration of PFOA, PFAS, PFNA, and PFHS on a subset of over 200 pair newborn babies. And meanwhile, working with the Emory Cheer and Targeted Laboratory led by Dr. Dean Jones, as well as the HERE Untargeted Metabolomics Laboratory led by Dr. Susan Sumner, we successfully completed high-resolution metabolomics profiling on both these maternal serum samples as well as these newborn dry blood spot samples. Then we use a well-established untargeted high-resolution metabolomics workflow to investigate the metabolic signature that are associated with PFAS exposure and adverse pregnancy birth outcomes in our cohort, including metabolomics, feature extraction, conducting metabolome-wide association study to figure out what feature are associated with the exposure or the outcome, and finally, followed by pathway analysis and chemical annotation, trying to tackle the underlying mechanisms. So using this workflow, 2 years ago, we published the first paper on Environment International that's led by our recent PhD graduate, Dr. Jerome Chang. And here, we mainly try to use targeted PFAS exposure assessment, high-resolution metabolomics among 300 participants within the Atlanta African-American maternal and child cohort. So here, as you can see, this shows you the eligibility for the enrollment in our cohort. And for this particular analysis, we mainly really focus on the PFAS impact on the maternal metabolome and the birth outcome during the early pregnancy. And working with the Emory Clinical Biomarker Cheer Laboratory led by Dr. Ding Jones, we successfully completed high-resolution metabolomics profiling on maternal serum samples collected during early pregnancy from 313 pregnant African-American mothers. So as for the analytical approach, we are following a meet-in-the-middle workflow, which is basically conducting 2 independent metabolome-wide association study, with one focusing on the single PFAS species and the other focusing on the fetal growth outcome indicators, where we're trying to identify overlapping pathway as well as metabolite that are associated with both prenatal exposure to PFAS as well as the increased risk of these fetal growth outcomes. And I just want to take some time to go over the results with you. So here shows you the association between PFAS and fetal growth. And in this particular study among pregnant African-American people, we observed significant association of maternal serum exposure to PFOA and PFNA with increased risk of reduced fetal growth. And as you can see here, both increased levels of PFOA and PFNA are significantly associated with reduced birth weight. And at the same time, that also with the increasing in the PFOA and PFNA concentration during the pregnancy, we are seeing a increased risk for gestational age. Now the question becomes whether or not we can use high resolution metabolomics in understanding the underlying mechanisms. So using a comprehensive meet-in-the-middle approach coupled with the metabolome-wide association study, we find out that the underlying potential mechanisms of the PFAS fetal growth relationship might result in a perturbation in several amino acids, lipid and fatty acid, and bile acids metabolisms, as well as sex hormones such as androgenic hormone disruption. So here shows you that there are more than 20 metabolic pathways that are associated with at least 2 or more PFAS concentration, as well as increased risk of reduced fetal growth, result in low birth weight and small for gestational age. And more excitingly, using chemical annotation, we are able to confirm 10 metabolites with level 1 evidence that are associated with both increased exposure to PFAS and increased risk of fetal growth. Specifically here, I want to use uric acid as an example. As you can see here, uric acid was identified as a potential intermediated biomarker representing the early responses of PFAS exposure and predicting the corresponding increased risk for reduced fetal growth. Specifically, as we can see here, one thing that based on our results, we can see that with the increase in the PFOA and PFNA, we are also seeing that the intensity for uric acid during the early pregnancy increases. And when the uric acid intensity increases, we are actually seeing that it's significantly associated with low birth weight, and it's marginally associated with increased risk for small for gestational age. And based on this result, we propose a potential hypothesis where that during the pregnancy period, with the increased exposure to PFAS, that might increase the oxidative stress. And once the oxidative stress is enhanced, there will be more uric acid being produced, which will result in placenta inflammation, oxidative stress, and impaired amino acids transportation, which then might result in reduced fetal growth. And this is just only one of the potential pathway, but due to the time limit, I won't go into details of all this, but we do observe several other amino acids, lipid, fatty acids, bile acid, as well as sex hormone could be added as potential intermediate biomarkers in the association between prenatal exposure to PFAS and reduced fetal growth. So to quickly summarize our findings in this first analysis, we found out that increased PFAS exposure was linked to risk of lower birth weight among babies delivered by African American. And we found out that the reason could be that PFAS might interfere the metabolism of amino acids, lipid and fatty acids, bile acids, as well as sex hormone, which might in turn affect fetal growth. And to validate these findings and further investigate the impact of prenatal exposure to PFAS on the newborn metabolome, our PhD candidate, Caitlin Tybalt, has recently completed a follow-up metabolome-wide association study as the last aim of our PhD study to characterize the molecular signatures of prenatal PFAS exposure and gestational age at birth outcomes in using the newborn dry blood spot metabolome. Here, we want to see if the metabolomics perturbation we observe in the maternal metabolome will be replicated and similar to those that observed in the newborn metabolome. And this particular paper was recently published in Nature Communications. So in this analysis, we focus on 267 mother and newborn diets within the Atlanta African-American cohort who completed assessment on both the maternal serum PFAS concentration and newborn dry blood spot metabolomics profiling. And in this study, we focus on 5 gestational age at birth outcomes, which include a continuous gestational age at delivery and 4 categorical outcomes, including preterm birth, early term birth, spontaneous early birth, as well as medically indicated early birth. So here, similar to our previous analysis, in this particular subset, we also observe significant association between PFOA and PFHS with early birth outcomes. Then we follow the semi-in-the-middle analysis where we're trying to identify overlapping pathway and metabolite in the newborn metabolome that are associated with both exposure to prenatal PFAS levels as well as gestational age at birth outcomes. And thanks to the UNC here Untargeted Lab Hub under the leadership of Dr. Susan Sumner, we successfully obtained about 7,000 metabolic signatures and signals in the untargeted metabolomics dataset after data pre-processing, quality control procedures, and data filling. And after using the metabolome-wide association study statistical modeling, when we focus on the newborn metabolome, we found out that there are more than 664 overlapping unique metabolic signals that are associated with both prenatal exposure to PFAS and adverse birth outcomes after adjusting for covariates, confounding factors, as well as multiple testing corrections. And we are able to confirm for these significant features, a total of 56 overlapping metabolites with high confidence ontology level 1 evidence. So this metabolite can be further broken down into the 7 major categories shown here. And I apologize that this font size might be too small for you. These 7 major categories really include metabolite that enrich in carnitines, bile acids, amino acids, and proteins, chemical messengers, redox reaction, fatty acid and lipids, as well as synobiotic metabolism. Specifically, something that I want to highlight here is that here perturbation in amino acids and protein seem to have the strongest effect associated with both PFAS exposure and fetal growth, with more than 11 metabolites enriching these 2 major pathways that are associated with both the PFAS exposure and the reduced fetal growth at gestational age. And at the same time, we also identified 14 unique overlapping pathways that are associated with both the PFAS exposure and gestational age at birth outcomes, with two metabolic pathways consistently showing the strongest association with every single PFAS indicator, including the urea cycle and tryptophan metabolism, which are closely involved in systemic inflammation, endocrine disruption, as well as oxidative stress. And I'm so very happy to report that for both of these metabolites and pathway, more than 80% of them also shows up in our previous analysis when we focus on characterizing the maternal metabolomics profile. So taking all these results together, we propose this potential molecular network that can maybe explain the toxicity of prenatal PFAS exposure on reduced length of gestation, which center around the metabolism and perturbations in the tissue neogenesis, in the neuroendocrine functions, as well as in the redox homeostasis balance. And we with future hypothesis testing study, we might be able to further figure out what is really going to happen in the human body when they are exposed to higher level of PFAS exposure, and how that molecular perturbation will contribute to increased risk of adverse maternal and child health outcomes. So to quickly summarize this second analysis, so in our Atlanta African American maternal and child cohort, we observed that increasing PFAS concentration were prospectively associated with a reduced length of gestation at birth, and that the newborn dry blood spot metabolome also reveal important molecular mechanisms and potential biomarkers that are associated with both prenatal PFAS exposure and gestational age at birth outcomes. And taken together, our study provides novel evidence and supports the current mechanistic understanding of how PFAS in utero will influence preterm birth and early term birth. And of course, our study for both these analyses have several limitations. First of all, like Dr. Liu just mentioned, in our study, only the individual effects of PFAS were included in this analysis, but mixture effects are possible. And also it's important to take into account to thinking about the joint effects of these multiple PFAS exposures. So as one of the next step, we try to look at using PFAS as a mixture to see their joint impact on the human metabolome. And of course, our limited sample size might also necessitate a larger scale study. And furthermore, the pathogenesis of the adverse birth outcomes is likely multifactorial, and it can be influenced by genetics, social status, and behaviors. And that's why we have recently submitted a grant that looks at the joint impact of exposure to PFAS, as well as social determinants of health and individual and neighborhood level experiences of racial discrimination, and try to see their joint impact on this maternal and child health. And finally, I just want to take the remaining time to highlight another very exciting project that we recently worked on. So instead of studying each of the PFAS species individually, we have started using both the advanced mixture analysis, specifically the quantity computation, with the collaboration with my collaborator, Dr. Stephanie Ike at Emory. And we are trying to investigate the joint effects of multiple PFAS species on maternal metabolome using QGCOM. And one of the take-home message is that if you look at the PFAS mixtures impact on maternal metabolome, we do see a similar pattern of metabolic perturbation that are enriched in several inflammation and oxidative stress-related amino acids metabolism that are associated with both PFAS, both single species PFAS, but also with the joint mixtures of PFAS. And moreover, for some of these metabolic pathway, they only seem to be associated with the PFAS mixture, which indicated that if we just use single species of PFAS to look at their impact on human metabolome, we might actually mix some of those very important cumulative effect or joint impact of PFAS mixtures. So just to quickly summarizing my presentation today, so we showcase that by using high-resolution metabolomics, we might be able to capture really sensitive molecular signals that are linked with PFAS exposure to internal dose and biological responses. We also observed that in this Atlanta African-American maternal trial cohort, prenatal exposure to PFAS might induce perturbation in both the maternal and newborn metabolome, centering around the perturbation of the endocrine disruption, oxidative stress, and inflammation, which might in turn increase risk of adverse birth outcomes. The exact mechanisms by which PFAS exposure impact maternal and child health still remain quite uncertain. So this needs a replication in other cohorts, and we have recently submitted an analysis proposal to ECHO that we are trying to leverage the ECHO here analyze multi-cohort metabolomics profile to see if what we observe among African American is also similar with other populations. And another important next step should be used of multi-omics and mixture analysis to get a more comprehensive and holistic view of really what's going on in the human body when we are exposed to higher level of PFAS. And validation of the findings using hypothesis-driven protocol in vivo and in vitro model are also very important because metabolomics per se is a hypothesis generation approach, and all the findings that we observe in this analysis need to be validated. And finally, hopefully taking all these findings together in the exposomics research, there will be one day for us to, based on these findings, to develop some sort of sensitive biomarkers for future solution-oriented and effective intervention to really protect the vulnerable population, especially among minority population and highly exposed mothers and children. So with that, I want to thank our funding agencies, specifically the ECHO program, as well as the NIH HERE program. And our group is currently recruiting postdoctoral fellow and graduate students. Feel free to contact us if you are interested in this work. Thank you.
Thank you so much for that wonderful talk, Dr. Leon. We really appreciate it, hearing about everything that you've been working on. We'll have time now, some time for questions. If you want to put your question in the Q&A, we can ask Dr. Leon. While we're waiting for some questions in there, I will ask a couple of questions I had. Given we just heard from Shelley about her work, I was wondering how the PFAS levels in your African American cohort, have you compared them to other racial and ethnic groups?
Yeah, that's a great question. So in our own cohort, we are seeing that compared to the enhanced level non-Hispanic Black, our cohort participants' PFAS level is quite comparable compared to the non-Hispanic Black. Compared to other race and ethnicity, we are seeing that some of the PFAS, including PFOA and PFNA, were slightly lower compared to the non-Hispanic Y and other race and ethnicity, but also at the same time, PFOS and also PFHS was slightly higher. And another thing I want to point out is that the effect size that we observe between PFAS and adverse birth outcomes is also very similar to the effect size that we observe among the ECHO-Y analysis among more than 5,000 participants that are led by Dr. Amy Padula's recent EHP paper.
All right, thank you very much. Here's a question from Erin Lebo-Skelly. She says, hi, so great to hear more about the important work you've been doing. Can you say more about potential solution-oriented intervention research that could follow up on these studies? Similarly, this is all very complex, and I'm wondering if any of these results have been reported back to participants and how they may have responded.
Yeah, thank you very much for raising this important question. So in terms of the solution-oriented intervention research, of course, one would be to try to think about how we can reduce exposure to PFAS, especially during critical time windows such as like pregnancy. So we have been not this particular analysis, but our colleagues at the Emory University has think about like solution-oriented research intervention that can potentially reducing them by, for example, increasing people's awareness of like what PFAS is, where they're coming from, what are the potential route of exposure, to try to reduce their exposure in their daily life, to, for example, decreasing the frequency of using non-sticky pen and other plasticizers that are important, or like contaminate drinking water that can potentially reduce people's exposure. And on the other hand, I think one of the great things about metabolomics investigation is that we might be able to have a better understanding of the underlying mechanisms that potentially maybe in the future we can develop some sort of nutritional interventions. For example, we are seeing a huge disruption of this amino acids metabolism and decreased level of several of these nutrients in the body. And perhaps with a hypothesis testing approach, we may be able to come up with some targeted amino acids that we can increase their amount during pregnancy through the dietary supplementations. But with all those future direction, we did need to invest more in the current study to test this out in clinical trials. And then as for your questions regarding the report back, we do think this is very important as an ongoing initiative as part of our Atlanta African American maternal and child cohort. And due to time being, I won't be able to go into details of it, but I'm more than happy to tell you more about that later.
Great, that's really great points. If I can ask one final question, there's nothing else in the Q&A, but I was wondering, as a toxicologist, if there are any comparable metabolomic results in animal studies. You mentioned in vivo or in vitro studies, and I don't know if there's similar studies with mixtures, but what can you tell us about similar results in animals?
Yeah, I think that's a great point, because like everything we see so far are hypothesis generation findings on cross-sectional analysis among a population. So some of, not our group, but our colleagues like at Emory or outside of Emory have done like great work in vivo and in vitro model, showing that some of this PFAS exposure does induce like perturbation in some of the similar pathway. But in those studies, they tend to use more like targeted inflammation biomarker and oxidative stress biomarker. I think one of the key future direction will be really try to do more like a targeted focus on this, for example, bile acids or amino acid and lipid acid pathway that we observe in the human, and try to focus on those marker in the cell line model and animal model, see if we can observe the same thing, and if there's any dose response relationship that we can establish. I think that's really key before we can establish any of these metabolic marker as a sensitive biomarker to understanding the impact of PFAS exposure on human health.
Great, thank you so much for your responding to those questions. We're at the end of our webinar today, and I believe there's a final slide about our next webinar in the next month, I think, sometime, which has not been determined, but there will be webinar 15 coming up. You can check out the HERE website, hereprogram.org. You can view previous webinars there and see updates on upcoming webinars. So I want to thank everyone so much for attending today, and the speakers and all of the questions and answers. Really had a great webinar today. Thank you all very much, and have a great day.